Welcome to part 2 of chapter 17, Innate Nonspecific Host Defenses. In this lecture, we're going to focus on chemical defenses, the molecules our bodies produce to inhibit or kill pathogens before they ever reach deeper tissues. So these defenses work hand in hand with the physical barriers we talked about in the first video, as well as the cellular barriers we're going to learn next. By the end of this lecture, you should be able to 1. Describe how enzymes in body fluids help protect us from infection. 2. List and explain the function of antimicrobial peptides, complement proteins, cytokines, and acute phase proteins. And finally, compare the 3 complement pathways, classical, alternative, and lectin, and understand where they converge. Chemical mediators are substances found in body fluids, tissues, and secretions that inhibit or kill microbes. Some are endogenous, meaning our cells make them, while others are exogenous, meaning our microbiota produce them. Some are always present at low levels, while others are upregulated during infection. And major categories include enzymes, antimicrobial peptides, plasma proteins, cytokines, and inflammation-eliciting mediators. This first section will introduce the chemical and enzymatic weapons our body uses, substances like lysozyme, lactoferrin, sebum-derived fatty acids, and others. Endogenous mediators are produced by our own cells, for example, sebum on our skin or lysozyme that's found in our tears and saliva. Exogenous mediators are produced by our normal or resident microbiota, for example, oleic acid on the skin or bacteriocins in the GI tract. Both types, endogenous and exogenous, will restrict or inhibit microbial growth, and their effectiveness depends on local factors such as pH, moisture, or nutrients. In our skin, sebaceous glands produce or secrete sebum, and that helps block hair follicle pores so pathogens cannot enter. We also have skin microbiota that will convert the sebum into oleic acid, and that lowers the pH of the surface of the skin and thus inhibits pathogen growth. So a stable, normal microbiome is key. If we happen to have reduced sebum or low humidity, that lowers our immune system. It lowers our protection because it becomes less habitable for our normal microbes to live there on the skin and will produce less oleic acid. This increases our susceptibility to pathogens. Within our digestive tract, several chemical defenses work together. Saliva, for instance, contains lysozyme, and lysozyme breaks down bacterial cell walls. You can see over here, it breaks down the connections or bonds between our NAG and NAM subunits. And lysozyme is most effective against gram-positive bacteria because they have those thick walls. Lactoperoxidase is also found in our saliva, and that enzyme produces oxidizing or oxidative molecules that will damage microbial membranes and enzymes. Esophageal mucus also contains lysozyme, and within our stomach we have a very low pH that can kill most microbes. Within the intestines we also have bile salts, different enzymes, and intestinal antimicrobial peptides that inhibit or kill microbes. And finally, we also have Paneth cells in the crypts of the intestines that also produce lysozyme. Within the urinary and reproductive systems, we have urine that provides a flushing action and also maintains a slightly acidic pH of about 6 that inhibits microbial growth. In the female reproductive tract, we have vaginal cells that produce glycogen, and then lactobacilli will ferment that glycogen to produce lactate, also known as lactic acid, that lowers the vaginal pH. And this acidic environment will suppress opportunists like Candida, as well as other pathogens. And the reason these lower pH environments work is because that most microbes, as we learned before, are neutrophiles. So that means they grow best at a neutral pH, usually between 6.5 and 7.5. So the most of the bacteria that would otherwise infect humans, they prefer neutral pH because human tissues and fluids are mostly near pH 7, and these pathogens have evolved to grow in the environments that they infect. For the microbes that can survive in more acidic or perhaps more basic environments, it's because they have adapted to those environments over time. Looking at the eye and ear, in our eyes we have tears, which contain lysozyme, and we saw earlier that breaks the NAG-NAM bonds in peptidoglycan. We also have lactoferrin, which binds to iron, and that makes it unavailable for bacterial growth. So it essentially starves microbes that require iron to grow. Within the ear, we have earwax, also known as cerumen, and earwax contains fatty acids. So that lowers that pH of the environment, helping to inhibit pathogens. So both tears and earwax will protect these surfaces that are frequently exposed to the environment. Within the respiratory tract, our nasal mucus contains many of the chemicals we saw earlier. We have lysozyme that breaks down those NAG-NAM bonds in bacterial peptidoglycan cell walls, lactoferrin that binds to iron to limit bacterial growth, lactoperoxidase, we saw that previously, and it generates oxidizing antimicrobial molecules that disrupt cell membranes and enzymes of microbes. And in the lower respiratory tract, we have surfactant, which can not only lower surface tension to help us breathe, but surfactant contains phospholipids and surfactant proteins that can insert into bacterial cell membranes to make them unstable. So surfactant especially is good at weakening or killing gram-negative bacteria. And together, these chemical defenses work alongside what we saw earlier in the previous video, the mucociliary escalator. Remember that there were goblet cells that secrete mucus along with cilia that can move the pathogens and debris up the respiratory tract where you can swallow them or cough or sneeze them out. The first checkpoint here asks, explain the difference between endogenous and exogenous mediators. So endogenous mediators are produced by human body cells. For example, sebum, lysozyme, stomach acid, and urine acidity. Whereas exogenous mediators, they're produced by our resident microbiota, not by human cells. For example, oleic acid is produced by our skin microbes. Lactate is produced from the vaginal lactobacilli or bacteriocins that are made by our gut microbes. So the key difference is endogenous mediators are made by us. Exogenous mediators are made by our microbiome. For the second checkpoint question, they ask, describe how pH affects antimicrobial defenses. So many body sites rely on low pH, which means an acidic environment, to inhibit microbial growth. For example, on our skin, that oleic acid can lower the pH, inhibiting pathogens. The stomach is extremely acidic, and that gastric fluid will kill most microbes. The urine is slightly acidic, and that can slow bacterial growth. And within the female vagina, lactate will also lower pH to inhibit microbes like Candida or other potential sexually transmitted infections. So lower pH can prevent pathogens from attaching, surviving, or replicating. All right, next we move on to one of the most important sets of non-specific defenses. These are called antimicrobial peptides, abbreviated AMPs. Antimicrobial peptides are small, broad-spectrum molecules that are produced continually or induced during infection, and they work by damaging microbial membranes, disrupting their DNA or RNA, or blocking their cell wall synthesis. And we'll find them on the skin, in the oral cavity, in the GI tract, and within various immune cells. Important antimicrobial peptides to know include defensins. These are produced by epithelial cells, macrophages, and neutrophils. And these disrupt microbial membranes. They can destroy bacteria, fungi, as well as many different types of viruses. For cathelicidins, these insert into membranes, causing pore formation and cell death in bacteria and fungi. Bacteriocins are produced by our gut microbiota, and they can inhibit competing bacteria. And we'll look at a few more in Table 17.3 on the next slide. Here is that Table 17.3, which summarizes antimicrobial peptide sources, where they come from, the target pathogens, and their mechanisms. So we saw these three already. Dermcidin comes from our sweat glands on our skin, and they disrupt the membranes of bacteria and fungi. And histatins are found on our saliva, and they specifically enter fungal cells, and they disrupt fungal mitochondria. So once they damage the mitochondria of these fungi, now these fungi can no longer make ATP, and that results in cell death. So these molecules together are essential for frontline protection. This checkpoint asks, why are antimicrobial peptides, or AMPs, considered non-specific defenses? It's because they do not target specific pathogens. Instead, they disrupt fundamental microbial structures that are shared across many different species. For example, AMPs can attack a wide range of microbes, bacteria, fungi, protozoa, even some viruses. And they work through general mechanisms such as disrupting membranes, damaging DNA or RNA, blocking the cell wall, or blocking some kind of intracellular function like damaging mitochondria. They do not target one specific pathogen, so their actions in general are broad spectrum. All right, now we're going to move forward and look at our plasma protein mediators. And as indicated in the name, they are found in our blood plasma. So plasma protein mediators are found in the blood plasma, and they will include three categories: acute phase proteins, complement proteins, and cytokines. These are mainly produced in the liver, and they will circulate in the blood and can rapidly amplify our immune responses. Let's start by looking at our acute phase proteins. These are made in the liver in response to inflammatory cytokines. So there are many different types that are described in Table 17.4 from our book. C-reactive protein and serum amyloid A have similar functions. They both coat bacteria, and that prepares the bacteria for ingestion by phagocytes. So this coating for phagocytosis is known as opsonization. And I have an image here that shows what that looks like. Here, let's say the circle is the bacterium, and you can see it's being coated. The structure is different, but let's pretend it's being coated by C-reactive protein and serum amyloid A. That allows our phagocytic cells to more easily bind to and take in through endocytosis the bacterium, the pathogen. Once it's taken in, the lysosome will fuse with that phagosome, and digestive enzymes will break down the pathogen. Our next set of acute phase proteins are ferritin and transferrin, and both of these bind to and sequester iron, kind of like a sponge. They soak it up, and therefore they are going to take away the iron that some pathogens need to grow. So that will inhibit microbial growth. And the last two acute phase proteins in our chart include fibrinogen, which is involved in the formation of blood clots that can trap pathogens. So here is an example of a blood clot. If you look at that meshwork, you can see fibrinogen has been activated to fibrin, which forms that mesh-like network that traps microbes. And we have mannose-binding lectin. Mannose-binding lectin is a liver protein that can bind to carbohydrates found on the surface of our pathogens. And that is going to induce the activation of complement proteins. We'll talk about these next. The complement system is made of over 30 proteins that are activated through 3 different ways. So these are always in our plasma and tissue fluids, but they become activated in these ways. So alternative pathway is when you have an spontaneous C3, so these are one of those proteins that becomes activated. In classical activation, the complement system is activated when antibodies in our blood detect foreign antigens. And then finally, in the lectin pathway, complement proteins are activated when mannose binding lectin binds to carbohydrates on the surface of microbes. For all of these proteins, and when they're activated, for these 3 pathways, all pathways will converge at something we call C3 activation. And we're going to see in a figure soon that activation will result in the generation of C3A, C3B, and downstream complement protein factors as well. This diagram shows how all 3 pathways converge. So here I see the alternative pathway, the classical pathway, and the lectin pathway. These are all different types of complement proteins, but the 3 pathways converge at C3, and I can see that breaks down C3 into C3b and C3a. Eventually, we're going to form C5b, which recruits these C6 through C9 complement proteins that form something called the membrane attack complex, or MAC. So the membrane attack complex punches through the cell membrane of that pathogen. Water will enter and the cell will swell and burst. In addition to forming that membrane attack complex, complement system activation can result in other outcomes as well. One example is opsonization. So I showed you this picture earlier for those acute phase proteins, like the C-reactive protein. It turns out that complement proteins like C3b can also coat the surface of pathogens, making them easier to phagocytose and break down. Activation of the complement system can also trigger inflammation. So C3a and C5a act as anaphylatoxins. And what these do is activate mast cells that are found under the skin and mucosa, and that will trigger the release of histamine, which causes vasodilation and increases the permeability of our blood vessels. In addition to that opsonization and inflammation, the complement system can also trigger chemotaxis. So this means movement. Taxis is movement. Chemo is toward or away from a chemical. So C5a, this complement protein, can attract white blood cells called neutrophils to the damaged area. So it acts as a chemoattractant. Chemoattractants, again, cause the movement of their targets, like neutrophils, to the area. And then finally, cytolysis is what we saw earlier. C5b activates the formation of that membrane attack complex made of C6 through C9 proteins, and that membrane attack complex looks like big holes in the cell membrane. It's especially effective against gram-negative bacteria since they have that outer lipopolysaccharide membrane. Our third type of plasma protein mediator are our cytokines, and these are chemical messengers that coordinate the immune response. They can act through autocrine signals. This is when the signal, the cytokine, actually binds to and affects the cell that secreted it, so the same cell. In paracrine signaling, the cytokines will bind to and affect neighboring cells that are nearby. Whereas in endocrine signaling, the cytokines will travel through the blood and affect distant cells. And these cytokines can also regulate cell growth, differentiation, apoptosis, which is sometimes called cell suicide or self-destruction, as well as chemotaxis. Here are those different types of chemical messengers. We saw autocrine, where the same cell secretes and receives that cytokine signal. Paracrine is when they're secreted to nearby cells, and endocrine is far away, so the cytokines travel through the bloodstream, through blood vessels, and affect a distant cell. We have three major cytokine groups. First, we have interleukins. These will regulate immune cell functions. Chemokines are cytokines that attract very specific white blood cells to the infection site. And interferons interfere with viral infection. So these are antiviral responses, and we have two subcategories here. Type 1 interferons, which are alpha and beta interferons, they're going to block viral replication and also induce apoptosis in infected cells. So this is when the cell self-destructs if it's been infected by the virus. And then we have type 2 or gamma interferons. These are going to activate additional immune cells to find, search for, and kill cells that have been infected by the virus. So these interferons do not save infected cells, but they will prevent the spread of the virus. So interferons are secreted by virus-infected cells. You can see the cell has been infected by the virus. And alpha and beta interferons are released and will signal neighboring cells to block viral replication and degrade RNA. They can also signal infected cells to undergo apoptosis, so the cell will destroy itself so the virus cannot spread. Gamma interferons will activate the immune cells, we'll talk more about these later, to kill infected cells as well. So again, interferons don't save infected cells, but their goal is to prevent the spread of the virus. All right, so our last group of chemicals are the inflammation eliciting mediators. Our inflammation eliciting mediators, their goal is to amplify inflammation. So these include histamine, which causes vasodilation, bronchoconstriction, and an increase in mucus production. Leukotrienes are similar to histamine, but they produce a stronger and longer lasting effect than histamine. So for example, leukotrienes can also bronchoconstrict or increase the permeability of blood vessels, but for a longer period of time and a stronger effect as well. Prostaglandins enhance inflammation and they can also contribute to fever. And bradykinins will increase the permeability of our blood vessels, and that can lead to fluid loss from the blood vessels into the tissues, which causes edema or swelling. Got 3 questions for this checkpoint. What do the 3 complement activation pathways have in common? So we saw that regardless of how they start, all 3 pathways are going to activate the complement protein C3 and eventually lead to that downstream formation of the membrane attack complex, or MAC. And all of them produce the same key outcomes. So these complement proteins can induce opsonization, remember coating the pathogen to make it easier to phagocytose. They can trigger inflammation. They can trigger chemotaxis to recruit white blood cells to the infected area, or cytolysis. They can break open the cells because of those membrane attack complexes. The second checkpoint question asks us to explain autocrine, paracrine, and endocrine signals. So for autocrine signals, these cytokines act on the same cell that released it. Paracrine is when the cytokine acts on nearby cells. And the endocrine signals, this is when the cytokine travels through the bloodstream to act on distant cells. Finally, the last one asks us to name 2 important inflammation eliciting mediators. So we talked about histamine that can cause vasodilation, bronchoconstriction or increase in mucus production, leukotrienes that are stronger and longer lasting than histamine, prostaglandins that can enhance inflammation and induce fever, or bradykinins that increase vascular permeability, which can lead to edema. So key themes of chemical defense include that defenses might rely on pH changes, such as a lowered pH, to inhibit microbial growth, different enzymes or protein cascades like the complement system. When I look at the complement system, we see a lot of tagging pathogens, recruiting other white blood cells, and destroying the pathogen. Cytokines are our communication network. So these are cell signaling molecules. And we're going to see that our chemical defenses work really closely together with the physical defenses we saw in 17.1, as well as the cellular defenses we're going to see in the next section. All right, let's recap 17.2. Describe how enzymes in body fluids provide protection against infection or disease. So enzymes in body fluids act as chemical barriers that damage or kill microbes. We saw lysozyme, for instance, in our tears, saliva, mucus, or produced by those GI tract Paneth cells. Lysozyme breaks down the NAG-NAM bond in bacterial peptidoglycan and thus lyses the cell wall of bacteria. That was especially effective against gram-positive bacteria with those thick walls. We saw lactoperoxidase, that's found in our saliva and mucus, that produces oxidizing compounds to kill microbes. And lactoferrin, that's produced in our tears, mucus, and saliva, that binds to and sequesters iron, that starves microbes that require iron. We saw that our gastric acid, so the pH of the stomach, is very low and will destroy most pathogens. And then we also have many different types of digestive enzymes in the GI tract that will break down microbial membranes, proteins, and nucleic acids. All right, for the second learning objective, they ask us to list and describe the function of antimicrobial peptides, complement components, cytokines, and acute phase proteins. So antimicrobial peptides, we saw they are small, nonspecific, broad-spectrum molecules. They kill microbes by damaging membranes or interfering with cell functions. So examples include defensins. They break down the membranes of bacteria, fungi, and viruses. Cathelicidins break down bacterial and fungal membranes. Dermacidin comes from our sweat and can break down membranes. We saw histatins. These are antifungal peptides found in our saliva. And bacteriocins are made by the gut microbes to kill bacteria. For the complement components, we saw that complement protein activation can lead to opsonization, coating microbes to make them easier to ingest and destroy by phagocytes. We saw anaphylatoxins that trigger inflammation. Complement proteins can also act as chemoattractants to recruit neutrophils, and they can form that membrane attack complex to lyse gram-negative bacteria. Cytokines, we saw these are the molecules that coordinate cell communication. We had interferons, for example, that interfered with viral replication. For acute phase proteins, these were made by the liver in response to inflammation. And I'll just do one example. We saw that C-reactive protein and serum amyloid A, both of those can bind to the pathogen surface and promote opsonization as well. For the third one, describe similarities and differences among classical, alternative, and lectin complement pathways. So all of them are going to lead to C3 activation, and that will lead to the activation and formation of that membrane attack complex. So ultimately, all of these pathways are going to produce the same outcomes. They're going to activate all of these different complement proteins and cause opsonization, inflammation, chemotaxis, and cytolysis. And that takes us to the end of 17.2 on chemical defenses. In the next part, 17.3, we'll continue with cellular defenses of the innate immune system.